Good morning, everyone. Before we get started, I would like to acknowledge that I'm speaking to you from Treaty 6 territory and the homeland of the Métis. We pay our respect to the First Nations and Métis ancestors of this place and reaffirm our relationship with one another. Welcome to the second session of the 2021 Plant Phenotyping and Imaging Research Center, or PERC Symposium on Flagship 3, Deep Learning for Plant Phenomics. PERC is funded by the Canada First Research Excellence Fund, or CFREF, and led by the Global Institute of Food Security on behalf of the University of Saskatchewan. My name is Mark Eramian, and I'll be facilitating this morning. I'm a professor in the Department of Computer Science, and I'm a co-lead of PERC Flagship 3 with Doctors Ian Stabness and Kirsten Bett. I'll start by filling you in on what to expect from this session. The talks from our speakers this morning have been pre-recorded and will play back-to-back, followed by a live Q&A once all of the videos have played. Please direct any questions you may have to the chat on the right-hand side. Once the Q&A starts, I will draw from these questions to spur on discussion with our speakers. I'm pleased to tell you that we have 5 speakers this morning. Our keynote speaker is Dr. Amy Tabb. Dr. Tabb holds degrees from Sweetbriar College, where she earned a BA in Math, Computer Science, and Music, Duke University, where she earned an MA in Musicology, and Purdue University, where she earned an MS and PhD in Electrical and Computer Engineering. She's currently a research agricultural engineer at a U.S. Department of Agriculture Agricultural Research Service Laboratory in Kearneysville, West Virginia. She'll be speaking on a tabletop system for 3D reconstruction of fruit form. Dr. Tabb will be joined in this session by Franklin Ogidi, and from the Department of Computer Science at the University of Saskatchewan, Masi Aslahi from the University of Saskatchewan Department of Computer Science, William Vanderkamp from the Global Institute for Food Security, and Lana Awada who is a Senior Policy Fellow in Food Security at the University of Saskatchewan's Johnson Shoyoma Graduate School of Public Policy. Immediately after this session, you'll have a chance to drop by the pages of our poster presenters to join their live video room tables and ask questions about their posters. This opportunity will run from 11 a.m. Mountain Time until 12:30. So now we'll start the pre-recorded videos, and I hope you enjoy the presentations.
Hello, I'm Amy Tabb, and I'm here to present some work on reconstructing fruit form with a tabletop system. So this is the first time I've presented this work, so this is brand new, so I'm really excited to share it with you all today. Before I get into the technical part of my talk, I wanted to explain a little bit about this acronym of where I work. So the USDA is the U.S. Department of Agriculture, and then ARS is the Agricultural Research Service, and the AFRS is the Appalachian Fruit Research Station. So this is what the place looks like from a drone view, and the lab and greenhouses are to the right, and in the center you can see the research orchard, which is attached to our lab. And at our laboratory, we work on temperate fruit problems, and there I'm a research engineer. So I'll explain how I got into working on reconstruction of fruit form. My prior work was on estimating the shape of trees, which is much, much bigger. So I met my collaborator, Mitchell Feldman, at a conference. So we met at Phenom 2018 on the last day, and it was like the last meal of the last day. And so Mitchell had been working on analyzing the shape of fruit in 2 dimensions with image processing, but he wanted to look at 3-dimensional shape. And at the time, he'd been working with laser scanners, and that was slow and didn't have really great results. So he wanted some more robust system for getting fruit shape. So we talked about some ideas for a project, and and then moved on from there. But one of the interesting parts of it is that we're on opposite coasts of the USA. So my location is in West Virginia, and Mitchell is located in California. So given all that, the research problem became how to robustly estimate this fruit shape using computer vision. And at the time, Mitchell was very interested in strawberry. And later we decided to explore, well, how does this system work on other fruits? And then we wanted to do so in a low-cost way, and a way that was modular, so that you could use a system in the field or the lab. And third element was that we had to do this in a way that worked for our 2 locations. So we weren't going to be flying back and forth. We had to be able to send the data back and forth. And so the accumulation of all that is that we had an abstract back in 2019, and then recently we were able to put this work together in a preprint, and so you can look at that if you're interested in more details. So I'm going to explain this tabletop system, but to do so, I'm going to give you some fundamentals of multiple view computer vision. So usually in multiple view computer vision, you're interested in getting some estimates of a 3D location of something. So that may be the shape or the points on a surface of an object, or you may want to know where things are in 3-dimensional space. So there are 3 key components of multiple view computer vision. One is camera calibration. That is the location of the cameras in space and their pose, so their rotations, and also some internal parameters. The second is image-to-image correspondences. So the link, the match of physical points in the real world to those pixels in the images that match those physical points. And then the third is the 3-dimensional reconstruction. Some people refer to those as, to that element as 3D models, but what that means is this estimate of some element of 3-dimensional space. So this could be a mesh or a point or voxels. So in general, if you have any 2 of these components, these essential components, you can solve for the third. And I have an asterisk there because there are many caveats with this, but that's the general idea. So, for example, if you had a pretty good camera calibration information and you had some image-to-image correspondences, then you can estimate the location of those points in 3-dimensional space or the 3D reconstruction. So in the context of this project, creating a tabletop system for fruit reconstruction, for the camera calibration step, we use an existing tool called Calico for estimating the camera calibration parameters. So that part is solved. For the image-to-image correspondences, we do segmentation of the fruit. And that's those segmentations in between the images. As long as you have camera calibration, you can match the points on the silhouettes as from image to image. So that gives us some image-to-image correspondences, and then combining those, we can create a 3-dimensional reconstruction using shape from silhouette methods. Now, the particular reconstruction you can create has some caveats to it, and I will explain those in the slides to come. This is the physical system that we use to acquire the images, and this was created by Mitchell out in California. So the way it works is that there's a mini computer here, and that when the user indicates, it triggers a transmitter and also the stepper motor. So the stepper motor starts to rotate a spindle, that has the fruit on it and these charupu tags. So the transmitter triggers this camera to acquire images. So the stepper motor is turning the spindle and the fruit's rotating while the camera is acquiring images. That process takes 9 seconds on average per sample. The cost of the system was about $1,600. And the advantages that we saw was that you could use this type of system in the lab or outside on a table. You know, when you are in a field where you have lots of samples coming in. The general size was about 2 feet by 3 feet. But again, this was in California. I didn't actually see the physical, or I didn't, you know, see it with my own eyes. I saw it in pictures. And finally, this design is a design that you could use with the software that we developed, but there are other designs that are possible that you could still use with the ideas we had in the paper. So for the camera calibration step, we use this other tool called Calico. And for that, we need to prepare the scene with these calibration patterns. So we have 3D printed cubes that are offset, and then we put on them some calibration patterns, and we don't need to know where the patterns are relative to each other. That is determined by the Calico method. And the result is that we know the location of the cameras with respect to the spindle. And so it's like simulating many, many cameras, even though in this case we have one camera. So the cyan pyramids are the representation of the camera pose with respect to the patterns, and these crosses are the patterns. So moving on to qualitative results, we tested this method on quite a few different products. And so here's some of those. This is the baby yellow potato and a strawberry, and one type of pear. Now these results are not to scale with each other, by the way. And here's a mini pepper, table grape. That was the smallest object, and another type of pear. And the Anjou pear was the one that had the most failures, but in general, our qualitative results seem to resemble the original products pretty well. So I'm going to talk a little bit about when things fail. So on the left is the raw image, and in the middle is the reconstruction, and on the right is a segmentation. So in this case, the segmentation didn't work very well for the Anjou pear. So when we designed the segmentation algorithm, we designed it to work generally for most of the samples, and we didn't fine tune it for any of the samples. So the Anjou pear happened to be one where the segmentation parameters didn't work for all of the pear. So I'm going to play that again. Okay, and then this doing reconstruction with silhouettes. There's some issues with that depending on what kind of shapes you want to reconstruct. So you can reconstruct many classes of shapes. For instance, mostly convex shapes you can reconstruct with silhouettes, provided you have enough silhouettes. You can reconstruct totally convex objects like that one. And you can even reconstruct objects with some singularities like points and saddle regions where the curvature is positive and negative in different directions. But objects that have concavities, like these big depressions, cannot be fully reconstructed with silhouettes, even if you have a large number of silhouettes from different viewpoints. So some portions of the object will be reconstructed, but not all of the object will be reconstructed. And I'll go into that a little bit more later. And there's a paper about the reconstruction method we used, which was which is a 2013 paper. So here's one of the fruits that we tested where we think that the reconstruction method is going to work pretty well because of the shape of the object. So it doesn't have any concavities except for the very, very small places where the seeds are. And unlike with the pear, the segmentation was very good. So strawberry was a fruit where we knew that it was going to work pretty well. And so here's sort of an intermediate case is the yellow pepper. So the yellow pepper is mostly got regions that can be reconstructed by this method, but it also has these regions on the side, these dimples. So as it rotates around, you can see those concavities, and the reconstruction does not represent those or doesn't reconstruct those. So that's sort of the middle of the road. Well, and the segmentation is very good in this case. So it reconstructs everything except the dimples. So if the dimples are very, very important to a particular application, you would need to do something different for the reconstruction method. So we had gotten some pretty good results for the qualitative evaluation, and the objects look like the 3D models look like the physical objects, but we wanted to assess, well, how well does this method do quantitatively, and that measuring three-dimensional shape is a very difficult thing to do. So we got some ground truth objects from Thingiverse, and a variety of them, these you've already seen. And then we printed them with a 3D printer. And we printed different sizes for each object to get some variety. And then to assess the difference between the ground truth object and the reconstruction from our system, we registered the reconstruction or aligned the reconstruction with the ground truth, and then assess the difference. And we did that in a couple of different ways, and I'll describe one of those ways. So this is that strange F-shaped object. So the segmentation has some errors, so you'll see some gaps here in the object, and see here there's an over-segmentation. I'll play the video again. So in this case, technically the reconstruction method should have been able to reconstruct this object very well, but there were some combined errors that resulted in some slight estimation error. And we found that over all the objects without concavities, the difference between the surface of the reconstruction and the surface of the ground truth, that root mean square error, was less than 2.7 millimeters. But then for the objects where we knew there would be some issues, those with concavities, we assessed those as well. So this is what those results look like. And where the concavities are, that's just in the reconstruction, that's just like a smooth flat section. And the root mean square error difference between the known model and the reconstruction, that was much higher as expected at 9.34 millimeters or less. So thank you very much for your attention during this talk. Some concluding points are that this was a low-cost modular system, and it had fast data acquisition. We had some good quantitative results for objects without the concavities. And in the future, we want to explore some different methods for the reconstruction so that we can capture concavities. So I look forward to your questions in the question and answer session. And if you have any other questions, you can feel free to email me.
Hi everyone, my name is Franklin and I am a master's student at the University of Saskatchewan under the supervision of Ian Stavnes and Mark Eramian. In this presentation, I will give an overview of the global wheathead detection dataset and briefly discuss some preliminary experiments we have done with it as part of my research. The global wheathead detection dataset is a large and diverse collection of high-resolution RGB images of wheat plants taken in the field. The first version of the dataset, which was released in 2020, had about 4,700 images and nearly 200,000 annotated wheatheads. In 2021, the dataset has been updated to include an additional 1,700 images and 80,000 annotations. The images on the slide here are a sample of the dataset, and it shows the diversity of the data set, including images of different genotypes of wheat taken in different lighting and environmental conditions, showing different wheat head densities and orientations, and even different planting practices. Such a large and diverse data set is not only important for exploring deep learning techniques for image-based wheat phenotyping, but it also enables researchers to develop robust solutions for a wide range of applications. The 2020 version of the dataset was born out of an international collaboration involving 9 institutions across 7 countries on 4 continents. The 2021 version featured contributions from an additional 8 institutions across 5 countries, with additional dataset from some previous contributors. The Global Wheat Challenge was introduced to crowdsource robust solutions to the wheat detection problem. The 2020 challenge was held on Kaggle, and it drew about more than 2,000 participants. Some of the annotations were cleaned up based on competitor feedback. This year, the challenge was held on AI Crowd, and it drew about 430 participants. In this year's competition, the training set consisted mainly of images from Europe, while the public and private test sets included images from North America, Asia, Africa, and Oceania. This style of data splitting encourages solutions that are more robust to domain shifts. It also led to the introduction of a new metric called the average domain accuracy, recognizing the fact that the sub-data sets all have different sizes. The 2021 dataset was also contributed to the WILDS benchmark project, which is dedicated to studying distribution shifts across many domains. For the next few slides, I'll discuss some of my work comparing supervised and self-supervised pre-training for wheat detection, and how the domain of the pre-training dataset affects transfer performance. The traditional transfer learning pipeline typically involves pre-training with a large dataset on an image classification task, and then fine-tuning on a different dataset, and sometimes on a different task. The supervised pipeline for this experiment is similar. So we first pre-train on an image classification task with the swing transformer backbone, and then we fine-tune for the wheat detection task using mass RCNN with a swing transformer backbone and a feature pyramid network. The self-supervised pre-training pipeline is a newer concept. It typically involves constructing an auxiliary task for learning representations of images without the need for labels. In a sense, it falls under the unsupervised learning paradigm. For the experiment, we used a self-supervised learning method known as MOBI. MOBI uses a swing transformer encoder in a Siamese architecture configuration and a contrastive loss function to learn image representations without labels. The fine-tuning step is the same as the supervised pipeline. At the pre-training step, we explored how the domain of the pre-training dataset could affect the transfer performance. Here we used ImageNet, which has 1.2 million general purpose images, the iNaturalist 2021 Train Mini dataset, which has 500,000 images of the natural world, and the plant subset of the 2021 iNaturalist Mini dataset, which has over 213,000 images covering more than 4,000 species of plants. And also the Sorghum 100 dataset, which contains 22,000 images of 100 sorghum cultivars. Notice that the dataset size is also a confounding factor. It's also a confounding factor in the experiment. Here are the preliminary results. Generally, pre-training helps compared to training from scratch. This is not particularly surprising, but it confirms that the self-supervised learning approach learns representations that are better than random without the need for a labeled dataset. Another general trend in the result is that transfer performance of the self-supervised pre-training method is either on par with or surpasses that of the supervised pre-training method, and especially when the dataset size is small. ImageNet pre-training is still the dominant approach, but this is likely due to the size and diversity of the dataset. On the other hand, the Sorghum 100 dataset, pre-training with the Sorghum 100 dataset shows the worst performance. But again, this is likely due to the small size of the dataset and the low interclass variability of the images in the dataset. The self-supervised pre-training method shows the most promise when using the planned subset of the iNaturalist dataset, where it outperforms both supervised and self-supervised pre-training with the full iNaturalist mini subset, even with 2 times less images. This suggests that a large and diverse plant or crop dataset on the same scale as ImageNet could potentially outperform ImageNet pre-training. With self-supervised learning, the dataset does not need to be labeled. In conclusion, the global weight head detection dataset opens up the space for research on robust weight detection solutions. And we have shown some promising preliminary results on self-supervised learning with planned datasets. Thank you.
Hi everyone, I'm Masi from University of Saskatchewan. Today I'm presenting our work in PERC Symposium 2021. Our work is about translating aerial agricultural RGB images to NIR spectral band. The importance of NIR band is to provide rich information in agricultural operations and experiments to determine crop parameters which are not visible to the human eye. However, collecting aerial multispectral images is more expensive than regular RGB cameras because multispectral cameras require specialized drones to be mounted on. We aim to employ an image-to-image translation model to transform RGB images to NIR spectral band. To do this, we propose a new data of matched multispectral aerial images for 4 crops across a range of growth stages. To collect our dataset, we use the MicaSense Red Edge, a multispectral camera widely used for agricultural purposes mounted on a drone. The drone is flown over crop fields and collects raw images. Each of the experimental plots represents different genotypes and species in a field. The collected data are raw images with spatial misalignment. To create our dataset, we employ a 2-step pre-processing algorithm to convert raw multispectral pixels to unitless images and align images pixel-wise. For RGB image translation to NIR, we use text-to-PIX network trained for 1,000 epochs. UNet and PatchGAN were used as generator and discriminator networks in our framework. The learning rate and loss function have changed in the original network. The paired RGB and NIR images are used for training and testing. To evaluate the performance of the model, 2 different experiments are conducted, including in-domain experiment, which tests the model with the crop of interest, and out-of-domain experiment, which tests the model across different crop types. We employed different standard measures like PSNR, SSIM, and SAM to evaluate the image quality. For the in-domain experiment, PSNR has reached to 31%, SSIM to 92% similarity for all crops, which show the generated NIR image is very close to the real NIR. SAM also shows a minimal distortion in a spectral band, like 3% distortion for all crops. For the out-of-domain experiment, PSNR hits 29%, SSIM 89% similarity, and SAM 5% distortion in the spectral band. These results also show that the model can generalize well to the other crop types. We also evaluated our results with specific domain measures such as vegetation segmentation over X-screen index and NDVI. X-screen exclusively uses RGB images, while NDVI requires NIR spectral band. To segment vegetation, we applied Otsu's thresholding technique across the X-screen and NDVI images. The results show that X-screen segmentation has a relatively close result with NDVI in mid-growth stage, while the X-screen is not similar to NDVI during crop's senescence period. This is because of the lack of green reflectance after senescence. However, the generated NIR is equally well as real NIR for NDVI-based segmentation. Our framework shows the efficacy of generating NIR band from aerial RGB crop images using our dataset. We also aim to test our framework with other crop types, such as corn or soybean, over different spectral bands. Thank you.
Hi, I'm William, and I want to talk to you about Plotvision, which is the Global Institute for Food Security's drone image analysis service for field scientists in agriculture. Before I get too far into it, I really have to acknowledge the huge number of people who have contributed to the development of this from the Plant Sciences Department at the University of Saskatchewan, the agriculture and agri-food researchers we work with, the Crop Development Center, the engineering department, everyone in the computer science department, and of course the Plotvision team themselves. So I'm going to quickly go over that. A really crucial part of it is Steve Shurtleff and Hema and their imaging team, who provide most of the images that we use in Plotvision. Couldn't do anything without them. And of course, they also do a lot of great research themselves. In addition to them, we work with the Crop Development Center with Curtis Posniak, who does wheat breeding, Sally, Christina, and Isabel, who do canola and carinetta breeding at Agriculture and Agri-Food Canada, Kirsten Bett, who does lentil breeding at the Crop Development Center. They provide the data we use, and they're the ones we're really trying to help. Couldn't do anything without them. And the computer science department, who we work with, and they do the real image analysis research that fuels what goes into Plotvision. So Kevin and Mark and Ian, of course, super important. And last but not least, the Plotvision development team. So, you know, everyone on here is actually working on developing this product, and I would be not going very far without them, that's for sure. So with all that being said, let's carry on. So what is Plotvision? Well, very briefly, our users gather drone images of their agricultural research trials. They upload those images to our service, and then we provide user tools and automation to turn those images into an actual data set, a per plot data set that can be analyzed and produce meaningful results, which we then output in the form of spreadsheets or various analytical tools. So you start by uploading. This is our dashboard. Every thumbnail here represents a different image capture mission. From there, once you're on a particular mission, you have to put bounding boxes around all the plots as seen in the top left. We try to automate that to some extent, we also provide user tools to make it easier for the user. And when that's done, the system goes away, automatically runs all kinds of analytics, determining things like the amount of flowering, the height of the crop, various spectral indices. And then we allow the user to analyze them in this plot workspace where they can kind of look at scatter plots, compare all the different plots, and so on. And they can also export the results in the form of simple spreadsheets, as shown here, or various vector and raster data that they can then import into their own GIS tools. So that's basically what Plotvision is. But now I want to talk about the progress we've been making. So this is just a chart that represents, okay, cumulatively, how much data has actually gone through Plotvision. And the blue line represents the number of missions that have had images uploaded. The red line represents the number of missions that have been successfully stitched, which means, through our photogrammetry pipeline, we've actually added support for every single multispectral RGB camera as of just fairly recently. So we're pretty proud of that. You can notice the number of missions analyzed is a little lower, but it's working its way up, and we're trying to provide the necessary tools to make it easier and easier for our users to get those data sets all the way through the analysis pipeline. Another way to look at it is you can look at the plot count. So the green line represents the number of total microplots analyzed, and that's gone way up because we've been analyzing larger and larger missions, which we never used to be able to do. Missions containing something like 3,000 plus plots. One way we've been able to do that is by doing plot propagation. So you can see here, this is an initial plot segmentation done early in the season using a lot of human input, moving the boxes around. Well, we automatically applied that to another mission of looking at the same trial. We just overlay those plots, those boxes, and it saves the user a lot of work. They don't have to do anything the second time around. Another thing that we're working on that also helps is we can really support these larger data sets with new tiling capabilities where you can zoom in and the resolution automatically adjusts, and you can dynamically go around the images and it'll kind of load itself as it goes. And this really helps deal with those larger data sets. So in addition to that, we're building in support for larger data sets, but what else are we doing to move forward in the future? Well, we're working not only with our local collaborators as listed here, but also other external groups like Nutrien, Nuseed, Corteva, and others. All of these groups have data in our system, and we're trying to support their needs, which is larger data sets, doing more advanced processing. One new advanced analysis we're working on is spatial processing. So you can see here a heat map showing, what are the differences in the results that we see, not only just based on the genotype, but also just the spacing of how the plots, where they're planted in the trial. And you can kind of see what looks like a spatial effect with the heat map. This is just beginning now, but we're hoping to automate that kind of stuff. Another really important thing is more and more advanced AI models accounting for the variety of data that can actually, that we can actually apply automatically to all the different data sets and all the different crops we're seeing. So one of them, really important, is unsupervised organ counting. In this case, we're, work by Teddy and Jordan, where they were able to highlight individual wheat heads, based on a data set that the model would have never seen before. And that's just an example of the kind of more advanced unsupervised work that we're looking to incorporate into Plotvision in the future. So that's my quick highlight of where we've been, where we're going, and I'm always looking for people to get in touch. So you can email me, william.vanderkamp@USask.ca, if you're an existing user and you have any questions, or if you're just looking to use the system for the first time. Love to hear from you. Thanks.
Good morning, everybody. Thank you for making time to attend my presentation this morning. Today's presentation is related to measuring greenhouse gas emission in the Canadian Prairies crop sector. We measure the contribution of land use to greenhouse gas emission and removals in the Canadian Prairies crop sector between 1985 and 2016. This includes the major greenhouse gases in the crop production, such as soil carbon stock as a net source or sink for carbon dioxide, nitrous dioxide emission from fertilizer application, residue retention and summer fallow, and carbon dioxide emission from fuel used on farm and for the transportation of outputs and inputs. Net greenhouse gas emission is measured by the total emission minus the soil carbon stock as a source of net sink or net source. Most data are obtained from Statistics Canada, and the environmental coefficients are obtained from previous studies conducted on the Canadian Prairies or from international sources. The results of the study indicate that net greenhouse gas emission decreased starting from the 1985 and continued to decrease in most of the years. The net greenhouse gas emission is the black line measured by subtracting the orange bars from the blue bars. The orange bars is the total crop production emission, including emission from fertilizer, summer furrow, residue retention, and fuel, and it decreased from 11 million ton in 1985 to 13 and continued to increase to reach 18 million ton in 2016. The reason behind the decrease in the net greenhouse gas emission is the increase in the soil carbon stock or carbon sequestration in the blue bars, which increased from 0.4, the negative sign indicate carbon sequestration, increased from 0.4 million ton of CO2 equivalent to 8 in 2005 to reach 16 million ton in 2016. The reason behind the increase in carbon sequestration is the adoption of sustainable technologies such as zero tillage and the replacement of summer fallow with crop rotation. Moving to talk at the provincial level, Alberta's net greenhouse gas emission decreased from 3.6 million ton to 2.4 million ton and to reach negative sign indicating a net sink in 2016. Saskatchewan net greenhouse gas emission decreased from 5 in 1985 to 0.8 in 2005 to reach 0.12 in 2016. Manitoba's greenhouse gas emission stayed at about the same level, 2.14 in 1985 and 2.1 in 2016. The reason behind that is that carbon sequestration in Manitoba was not high enough to overcome the increase in the crop production emission. And this is because the province is continuously use tillage operation to decrease the humidity, the precipitation in the province. We find that the adoption of sustainable technologies can reduce greenhouse gas emission. The greenhouse gas emission decreased by 80% between 1985 and 2016, and by 57% between 2005 and 2016. This significantly exceeded Canada's commitment to Paris Agreement, which targeted at the beginning 30% and then 50% below the 2005 level of emission by 2030. There are three major areas that require research-based policy responses, including tillage practices in Manitoba, fertilizer application rate, and fuel use for the transportation of outputs and inputs. Well, there is some hope. The introduction of sensor-based digital data, the Internet of Things technologies, big data analysis, and smart farming and digital agriculture are promising to provide farmers with a technique to improve their decision-making and the efficiency of input use, promising to provide solutions for climate change, food insecurity, and improving agriculture productivity and profitability. However, there are lots of questions related to the benefits behind the data-driven technologies, the rights of farmers, the role of regulation intervention, and knowing that there are many stakeholders are going to be involved in the design and the design and the implementation of climate change policies. Thank you very much. If you have any question, please don't hesitate to ask.
Hello again, everyone. Hope you all enjoyed the great series of presentations that we just got to see. We've now come to the Q&A panel portion of our session today. So I would like to invite and encourage all members of the audience. If you have a question, please drop it into the chat and that will be fed to me and I will pose the questions to our panelists. Our first question today comes from Peter Lee of the University of Saskatchewan. And this question is for Amy. And the question is, which research areas could the 3D reconstruction of fruit form be used in?
Okay, so the original motivation for the work was for a strawberry breeding program. So appearance in strawberry is very important, particularly in California, because they package the strawberries and then ship them out across the US. So they wanted to characterize the shape of the fruit form, and that's difficult to do in 3D space. And then it can also be used for phenotyping for different crop treatments of fruit. So you may know that a treatment affects the fruit shape in a certain way, but you're not sure exactly how it affects that treatment, how it affects the shape. So then you can characterize exactly how it affects shape. Yeah. So, and we talk about this in the paper, but it can, this type of system, the turntable style system, could be used for things that are sort of small, but you'd have to scale it up if you want it for big things like melons, for instance.
Okay, thank you, Amy. And the next question we have is also for you, Amy, and it comes from Ian Stavness from the University of Saskatchewan. And Ian asks, what are some of the challenges with transferring the tabletop system to the field? Would it be possible to generate 3D fruit shapes in situ?
I guess it depends what the goals are when you ask that kind of question. So like what type of resolution do you want and what kind of accuracy do you want? So the challenges would be to get those three components that I listed. So the camera calibration part and the correspondences and then the reconstruction. So for the camera calibration part, you could construct a system with multiple cameras because doing a turntable in situ is not going to work. But the challenge in the field is getting enough views of the fruit. Especially with apple, we have a lot of leaves that cover up the fruit. And then, so you may be able to get partial reconstructions and not full ones, but of course it really depends on what crop you're talking about and what kind of leaf coverage it has. So, so yeah, there would be a lot of challenges, and it really depends on what your goals are.
Okay, thank you very much. Next question comes from Bobby Helgeson, and this question who is from the University of Saskatchewan, and this question is for William. You mentioned that you've been working with external stakeholders on spatial mapping of plots. This is really interesting and relevant to below ground processes. Has this same type of mapping been done on any of the PERC plots?
Thanks for the question. I would characterize our spatial mapping work as very much in the early stages, and this is the kind of thing that we have, whether we're working on the external data or the internal data, there's no real separation there on, like we're going to work on all of it for that kind of work. But I think the most important thing is if you're interested in it, we should definitely chat about it and find a way to work on the kind of data that you're interested in. But yeah, it's still pretty much in the early stages there, and we haven't really had a significant result yet, either on the internal or external data.
Thank you, William. I see we're already out of audience questions. So while I encourage the audience to bring in some more questions, I'll ask maybe one or two of my own questions here. I'm going to move to Franklin. Franklin, I noticed that in your table of results, you had a lot of different measures. And I was wondering if you could take some time to interpret that table for us a little more. For example, you have 3 different measures. You have MAP, MAP 50, and MAP 75. And I'm wondering if you could elaborate on why you need all those different measures and what they tell us about the training performances under the various conditions.
Thanks for the question, Mark. The ADA metric was used as part of the competition, and that's why I included it there. The MAP metrics are standard for object detection models, and the MAP are measured at different intersection of our unions. So the MAP 50 will be measured at intersection of our union of 50%. And then the MAP 75 will be measured at an intersection of a union of 0.75 or 75%. So as the intersection of our union increases, is a more stricter metric. So it's basically saying that the predicted box has to match closer and closer to the ground truth box. I don't know if that answers your question.
Yeah, I think it definitely does. How do you use what is the purpose of having those measurements at different levels of IOU though? You got the 50 and the 75. What is the purpose of investigating both of those, if you don't mind me asking?
One reason would be to find out how good the predictions of the model are. So if you have, so for example, pre-training on self-supervised learning versus supervised learning, you can tell based on those stricter metrics, you can tell which model is basically a better model.
Okay. Thanks, Franklin. I'm going to ask a question of Masi. Masi, in your second last slide, you had I believe you had a comparison of the segmentations that you got from the predicted NDVI image and comparing them to the segmentations you got from an excess green image. On the same slide, you also had examples of the segmentation that resulted from the real NDI images. So I'm wondering why you were comparing the predicted segmentation to the excess green as a baseline instead of comparing it to the segmentation from the NDVI, or have I completely misunderstood?
Thank you for the question, Mark. We compare actually NDVI, real NDVI and generated NDVI with excess green because we wanted to show what is the importance of capturing NIR images compared to just RGB image for segmentation, because as we know, RGB image cannot capture everything, but in terms of invisible spectral band, NDVI, I think it provides more rich information for breeders and farmers. We compare that with excess green to show the difference between segmentation with excess green and NDVI, but in terms of real NDVI and generated NDVI, we wanted to show how close are the generated NDVI to the real NDVI, generated NIR to real NIR, and our model can generate very good quality images.
Okay, so you did actually compare the NDVI predicted the segmentations resulting from the predicted NDVI to the segmentations resulting from the real NDVI?
Yeah, actually, in that term, we show that the generated NDVI and real NDVI has a similar result compared to excess green, which shows that the generated NIR and real NIR are very close together.
Okay, thank you. I think next I will ask a question of Lana. You mentioned that the decreases in prairie greenhouse gas. Well, the whole point of your talk was you showed that decreases in prairie greenhouse gas emissions from farming have exceeded the Paris Agreement targets. What I was wondering, keeping in mind that I'm a computer scientist, how big of a component of the overall net greenhouse gas emissions in Canada does this represent? And how much does this contribute to reducing Canada's overall net greenhouse gas emissions?
Well, agriculture in Canada accounts for about 10% of total greenhouse gas emission in Canada. The crop sector, which I'm measuring here, accounts for about 6.5%. What's happening on the prairie captures about more than 80% of Canada's arable land. So that's the importance, of decreasing greenhouse gas emission on the prairies, because the prairies has more than 80% of Canada's arable land.
Great, thank you for that context. Now I can really appreciate why that's so important. Thank you. Looks like we have a few more audience questions. The next question I got is again for Lana, and it's from Bobby Helgeson at University of Saskatchewan. She says that you showed that for Saskatchewan, the net change in greenhouse gases primarily occurred before 2005 due to the early adoption of reduced tillage. Since that time, our net gains were negligible. What does this mean for our current initiatives that work to build carbon opportunities in the agriculture sector based on soil carbon storage and sequestration?
Well, the main reason for this reduction is the adoption of zero tillage, which promotes carbon sequestration, and the replacement of summer fallows crop rotation. So in general, we have more cover on the soil, which increases crop soil sequestration. The reduction goes in parallel with the adoption of zero tillage, but we also identified increase in the emission and the total emission. And we have two major problems that we identified, which are the fertilizer, and particularly the low efficiency of fertilizer uptake and fuel for transportation of inputs and outputs. So for the future research and policies, we believe these two areas need more research, and also the tillage operations in Manitoba also was very low. The adoption of zero tillage in Manitoba is not more than 20% over the whole period of study. I don't know if that answered Bobby's question, but if not, please, Bobby, ask again.
Well, in any case, Bobby is more than welcome to submit a follow-up question. The next question in the queue is for William, and it comes from Venkat Bandi from the University of Saskatchewan. What has your experience been like in scaling Plotvision from an internal researcher focused tool into a SaaS tool for external corporate clients like Nutrien?
Hi, thanks for the question. I think the first thing to emphasize is that actually Plotvision is still very much focused on the PERC researchers and the internal researchers here at the University of Saskatchewan and Ag Canada. Like for instance, in respect to the other question I got about spatial mapping, actually the main impetus for that has come from the researchers in PERC, and that's like the primary data sets we're working on for many of the research projects that we're doing. with that being said, yes, we've been starting to branch out to various external groups, and everyone has different needs. I'd say overall, there's two main things we've noticed is that as we have more users, our system has to be more and more robust. We have to be much stricter with our deployment cycles. Like in the early days, I would just kind of take down the system every now and then when I wanted to make a change, and that's not really like something you can do anymore. And I guess another really important aspect is basically just the size of the data sets. And it's not necessarily corporate clients versus non-corporate clients. It's just basically who are the ones with the 3,000 or 5,000 plot breeding trials that we need to support with high resolution image data. And that's been the real challenge, is scaling up our system to handle large data sets in a way that isn't gonna cost us any more performance, isn't gonna cause any crashes. So that's what we've been really focusing on for the last half year or so. Hope that answers the question more or less.
All right, thanks, William. Where are we? Okay, for the next question is for Amy, and it comes from Andy Sharp from the Global Institute of Food Security. Do you have plans to carry out these reconstructions across genetically structured populations, say in strawberries, where shape variation could be segregating to further assess the robustness of the reconstructions?
So this the work that I presented to you was about the method for capturing the reconstructions, but then now that we've gotten sort of a understanding of how the method works and how well it works and how it doesn't work, then the next step would be to use it in a proper experiment, and I would be doing that with biologists. So it really depends on who I'm working with and what they decide they want to study. So it may be strawberry, or a lot of people at the lab where I am are working on apple and pear.
Okay, thank you. Next question is for Lana, and it's from Ian Stavnis at the University of Saskatchewan. You nicely showed the impact of different tillage practices across the prairie provinces. Do you think that regulation would help increase zero-till practices in Manitoba, or would you recommend a different approach?
I was answering this message, on comment. Well, I probably recommend a different approach. Manitoba has problem with high precipitation and flood especially over the last 10 years. And farmers were forced to till the soil to dry out the land and prepare it for seeding in the spring. But also Manitoba adopted crops that are heat lovers, such as potato and soybeans. And farmers were also tilling the soil to grow this type of crops. I'm not sure if reduced tillage, will help Manitoba. Probably a different approach. I don't know what it is, but Manitoba has problem with precipitation, and farmers are obliged to till the soil to dry out the land. So probably different approach, or combination probably of reduced tillage and something else. So we need research in this area. That's what we identified, that more research is needed in this area.
All right, thank you. Next question is again from Ian Stavnis, this time for Franklin. Many people are familiar with convolutional neural networks. Could you explain more about transformer networks and how they are different from CNNs, convolutional neural networks?
Thanks for the question, Ian. Convolutional neural networks use filters to create feature maps of images and then reduce those feature maps to a feature vector and then use them for various tasks. Transformers, however, were born out of, they were originally developed for natural language processing. So a task like translation of one language to another. Transformers work by taking, for example, in the natural language domain, it would take the words as tokens and then do something called a self-attention, multi-headed self-attention with the words. So what that means is if you're looking at a particular word, if you're trying to translate a particular word, you look at all of the words in the sentence and calculate some sort of probability or probability distribution of how those other words are related to the word you're looking at. And then use that to make a decision about the current word. So this same principle is applied for vision transformers in the vision domain. So you take an image and you break it down into patches, and those patches form the token. And then you apply the multi-headed self-attention. So basically you have in the normal vision transformer, you have a global view of the entire image. So if you're looking at one patch, you compute how it's related to every other patch in the image. The swing transformer we use for these studies is interesting because it's a modification of the vanilla vision transformer to include some form of hierarchical feature extraction. So in convolutional neural networks, as the network gets deeper, you can introduce more features to learn more, introduce more filters to learn more features. The swing transformer does the same thing. So as the network gets deeper, it introduces more multi-headed self-attention layers. Basically, it learns more channels in the channel dimension that it can use to, in the downstream part, to do the classification or object detection task.
Thank you, Franklin, for that very detailed answer. I think I'll slip in a similar question for you, Franklin, that I had. For the folks in this virtual room who are not computer scientists or deep learning experts, could you also elaborate on why self-supervised learning on data sets without labels works and what that self-supervised training phase is actually learning?
Okay, so the whole point of doing deep learning is to learn feature representations from raw data. And classically, we use labeled data to kind of inform that learning process. So with self-supervised learning, the main advantage is that you don't need any labels at all. And it works by doing some sort of, like I said, a pretext task where you construct a task that you don't really care about the performance of the model on, but all you care about is learning representations, useful representations that you can use later. So for example, you can do like, you can take an image, rotate it a bit, and then try to predict the rotation. That's an example of a pretest task. You don't need to know that what's in the image is a cat or a dog. You just need to know that you applied a 90-degree rotation and you try to predict that. So the type of pretext task we use here is contrastive learning, and it's kind of related to deep metric learning, where imagine you have an embedding space where you have all the pictures of cats and all the pictures of dogs. You want to cluster all the pictures of cats together and cluster all the pictures of dogs together, but you want them to be separate. And that's what contrastive learning does. So it tries to build a kind of embedding space that clusters pictures or images of similar classes together and pushes apart images of different classes.
Great, thanks, Franklin, for that explanation. I'm going to move back to William for a moment. And I'm going to ask William, do you have any, I hope I'm not putting you on the spot here, but do you have any success stories of actual users that are using Plotvision to analyze data for crop breeding?
So we've had a number of breeders who have been using the system to actually, get metrics out, and, they've successfully produced metrics over time for all their thousands of plots. And we produce, a variety of metrics like crop height and various spectral indices, and we incorporate vegetation segmentation. So all those numbers have been produced for a couple of different groups. With that being said, I think we still have to reach sort of the light bulb moment where we're really able to take those numbers and apply them to the breeding cycle in a meaningful way, where the selection decisions have been really driven, and the value has been improved, or the costs have been saved, like in a really significant way as part of the whole breeding cycle. So I think we still have to get there. We definitely do have success stories. I think with our, maybe the best way to put it is that we've got a couple of paying clients this year, and we've been able to produce data for them that's acceptable within the contract and so on. But with that being said, it's still kind of exploratory. It's still kind of, okay, what are we actually going to do with this data that's going to be really helpful and really driving value?
Thank you, William. And following on from that question, and again, I hope I'm not putting you on the spot, but what is the most exciting feature that you've discussed adding to Plotvision, in your opinion, that you think is possible, but you haven't been able to do yet? What can you say anything about what exciting features might be coming down the pipe? Maybe you can't, I don't know.
Yeah,
but
no, we've definitely got some plans, and it's part of what I highlighted already, which is that we're trying to automate all the post-processing steps that you would do on your metrics. Like, it's one thing to compute metrics, like all the metrics that I mentioned, but it's another thing to normalize them all, and and basically do that kind of spatial normalization, for instance. I think is going to be really important. Really excited about that. And as I said, I'm also really excited about incorporating more AI. And as a number of the talks here today were basically about generalizing, like Franklin and Masi both talked about generalizing to a variety of different data sets. And a real big struggle we've had at Plotvision is that we try to automate the whole analysis process. But there are so many different data sets with different cameras and different crops that it's been really tough to do that robustly for anything very advanced. Like, we can do well on our relatively simple metrics, but if we're doing things like counting organs, for instance, it's much more difficult. We haven't been able to do it yet in a general way, at least not in a way that we've incorporated into our system. So that's why you see the exciting, I think the thing I'm most excited about is the research work that's been presented today, frankly. And I hope that we can incorporate that in a fully automatic way into our system.
Well, that's great. Thanks. I'd like to go to Masi for a minute here, and I've got a couple of questions. I've got a question or two for Masi that I hope aren't too technical in nature, but I'm going to try asking this one. In your second last slide, you had some quantitative results about the overlap between the segmentations of excess green, the predicted NDVI images. And you gave some numbers as percentages, that you gave the overlap as percentages of 85% and 75%. And first, I just want to confirm those are averages, right, over your data set?
Yes, those are average numbers across all data sets, because we used 4 different data sets.
Okay, great. And do you have any sense of how much variance is in those averages?
Actually, results for data sets were ranging maybe from, I can't actually remember the exact number, but maybe actually ranging from 75% overlap to around 88 or 90% overlap. Because some of the data sets has actually many genotypes, like canola data set, and there are actually too many varieties of canola crop in the data set. But yeah, actually that's the average across all data sets.
Okay, so that's the average. And then you did observe some some data sets where it did considerably better and some where it did considerably worse. And so I guess a follow-up question would be, do you have any sense of what was causing those differences in performance? Like if you had the 92% data set that you mentioned, what was it about that made it easier, or the one that was 75, what was it about that one that made it more difficult? Do you have any insights into that?
So actually, we have, I think, 4 data set, lentil, barley, canola, and wheat, but we combine lentil and barley together in a pulse data set. The number of lentil images was very low compared to the other data set, and that data set was very imbalanced. But still, the model can actually recognize the features and plant phenotypes of lentil. But that could be one reason because of the imbalanced data set, and that caused the problem of actually a little deficiency. And also canola, because it has several different features like flowers and leaves or the other actually phenotypes. That was another reason for, I think, that low accuracy in canola. Wheat also has many tiny phenotypes, but the model couldn't recognize those small phenotypes. But still, the model, the accuracy of the overall actually performance was acceptable and in a promising way.
That's great. Thank you for those insights into the things that were making it more difficult for your model to learn. That's really interesting. I'm going to go to a question for Lana from Andy Sharp. And Andy says, ditto to Ian's comment. It was nicely compelling way to present how things have moved toward a net zero and level, and also indicating some key areas to further improve things, including at a research level. Will you be developing this into a policy recommendation document, or has that already happened?
Well, we have a paper that shows the measures, and we share also all the data. The data is at the soil zone level, climate soil level. So it's detailed data since 1985. The paper is submitted to PLOS 1, and it's in final revision now. In the paper, we concluded the paper by providing a policy recommendation and future research recommendations. We also, related to fertilizer, we also wrote a policy brief related to the problem of fertilizer, especially the low intake of fertilizer efficiency. Currently, we are, as one of the solutions is data-driven technologies, but I'm an economist, so all my problem now is, the data-driven technology services companies are acting as monopoly and monopsony at the same time. So I'm looking at the distribution of the benefits from adopting data-driven technologies by farmers. So that's what I'm working on, still in the early stage of research. So that's what's happening related to this work.
Great, thank you. Let's see where we're at. I'm going to come back to Amy, and I'm going to ask her a little bit more about the nature of your segmentation algorithm. You noted in your talk there that there were some problems with the segmentation for certain shapes, and I'm wondering if you could elaborate a little bit on what about the algorithm was causing those failures, or what about the imaging conditions that were causing those failures, and how you might mitigate that, or if it's even necessary. So what would be the next step there in that for the segmentation algorithm?
Okay, so first, the segmentation algorithm we used was a pretty basic one. So we used a background subtraction technique. So we modeled the darker parts of the background and the lighter parts of the background, and colors that were significantly different from those two were counted as fruit. So with the lighting in that data set, that we used in the paper, there seemed to be more illumination on the top part of the fruit versus the bottom. And there was only one fruit that seemed to have problems, which was that particular pear cultivar. So going forward, if you were to use the same algorithm that I used in the paper, you could tune the parameters for your particular crop that you're working on and get a variety of examples to make sure that it's going to work with the range of colors that you're going to have. Another option would be to use deep learning. If you want, the downside of deep learning is you need training data. So, but that is, yeah, another option too, that might work really well.
Yeah, I think that matches my intuition of what was going on. Yeah. So like when I was looking at it, I thought that the problems were often appearing in areas where there was more shadow from the lighting, and so then the colors were darker, and that was overlapping with your modeling of the dark background. So do you think you could solve the problem with a more uniform, diffuse lighting conditions? Would that be another way to solve the issue?
Yes, possibly. We acquired the data, and then back in January of 2020, and then we did the analysis in 2021. And really, we wanted to just assess how would a method do in general, and not tune it, or it's like over tune it to get really great results, but just to see like how does this do in general. But yeah, you could do different things. Of course, it would depend on the shape of your fruit. So again, it depends on what crop you're going to be working with.
Okay, thanks. Another question for Amy from Ian Stavnis. I like that your tabletop system is inexpensive and modular. Do you have any plans to replicate the system in other labs or translate it into a product or service?
Okay, so we have a version of that system in the lab in West Virginia now. So the machinist who works with me, Scott, he created one for a different project. And then his actually has a tabletop versus a spindle, but then we're going to put one of these 3D printed cubes on it. And then you don't need to know the relationship of the patterns to each other. So then you just tape the patterns to the cube. So it's very low tech to put the patterns on there. So Scott's design is very different from Mitchell's design, but it's going to work essentially the same. And then translate it into a product or service. So in my position, I don't really have control over that. So if a company wants to commercialize it, then they could contact me, and then we would go from there.
Thank you, Amy. I'll just say that I'm running pretty low on questions, so this is sort of be a last call for questions from the audience, or we're going to run out of questions and we'll be ending the session. Okay. So I do have a question here from Venkat. Sorry, your last name escapes me. It wasn't Venkat Bandi. There you are. This is a question for William, and he says, does Plotvision use different models for different plant varieties? Is there an automatic pipeline where the model varies based on the type of plant, or is it up to the breeder using that tool to manage that?
Hey, thanks for the question. So the way we designed Plotvision was basically to be this big database that keeps track of everything. So the user really only ever has to enter one piece of information once, and then they'll be in our system. All the data will be linked up in the right way. And basically we can be smart about our automatic processing. So what that means is, when the user first when they first enter in, a breeding trial that they're doing, they're going to give us, what's the crop they're working on. And they're going to give us like all that relevant high level metadata. And then we'll hold on to that. And then when they upload images, we're going to use all those parameters, like what is the camera, what is the crop, what's the GPS location, what's the height, what's the resolution, and all that is supplied as a parameter to our fairly flexible internal processing automation pipeline, which basically takes all those parameters and then provides them to the various models that are each going to be producing results. And yeah, basically the idea is that we can kind of plug in different analyses based on, you know, what parameters are available. And it's supposed to be all modular and all automated. And so what we've been able to do with this kind of like total automation approach, which does have its downsides, what we have been able to do though is for data sets that, you know, clients would typically wait months before they get any results back, you know, it'll be through our system and out the other end in under a day. So that's the kind of benefits that we can get.
Okay, thank you. Thank you, William. I'm coming to the last of my prepared questions. So again, audience, if you have any burning questions you'd like to ask before we reach the end of the session, now would be the time. So my very last question that will come from me is once again for Amy. And I was wondering about how severe of a problem is it that you can't detect certain concavities? So do you have any sense of how that inability to detect and properly render certain types of concavities, how that would affect the downstream phenotypes that you're able to analyze, and whether this is a problem that needs to be handled in some way or not?
Okay, so yeah, it's an interesting question, and I feel, I'm sorry, but I'm going to give the answer I give a lot, which is it really depends on the research problem from the biologist. So if you're studying apple and you want to know about the stem area and say if there are stem pools, if that's a cultivar that has a lot of stem pools or some selection, then there's a concavity there. So you may really care about this region. So it really depends on what is the research question, what kind of answers do we want from it, and then, okay, are concavities part of that answer? And if they aren't, then, okay, we don't need them. But if they are, then you need to incorporate them.
Yeah, so obviously, yeah, thank you for that. Obviously, you know, the concavities from the silhouette, it's a tricky problem. Can you envision, if you needed to better render the concavities, can you, maybe this isn't a fair question because I know it's hard, but do you see any way that you could improve that? Do you envision any solution to that at all that doesn't drastically inflate the cost or the complexity of the system?
Yeah, so from the reconstruction from silhouettes, then you can use that as an initial solution with doing multi-view stereo or something like that. So that's what people have done through the 2000s, is they use this shape from silhouette as the initial solution, and then they could refine that with stereo to deal with other parts. So that's what we want to work on next.
Yeah, stereo, that makes sense. Thank you very much. Okay, so I'm out of questions, and we have no questions in the audience queue. So I think we're going to, a last minute question just arrived. This is a question for Franklin. It is from Vyom Patel from the University of Saskatchewan. And the question is, how is the generalizability of the self-supervised approach compared to the supervised approach across different phenotypes and crops? So comparing the generality between those two approaches. The generalizability between those two approaches. Supervised approach can sometimes completely fail or require training from scratch when tried on different types of images. So does this issue carry over to the self-supervised approach, is the question.
Thank you for the question. Unfortunately, we haven't really tested the generalizability of the self-supervised approach yet across different crops. We're currently working on that.
is this type of generalizability question been studied by anyone else in any other domains?
Yeah, there have been studies in, if I remember correctly, the natural world domain. So, for example, using the iNaturalist dataset and then as a pre-training dataset and then transferring on multiple different downstream tasks. So right now, the self-supervised approach and supervised approach are very close. So sometimes it's like 1% or 2% difference at most. This can be positive or negative. But the main thing to note here is that the self-supervised approach does not require a trained labeled dataset. So for an unsupervised approach to come close to a supervised approach, I think that's promising.
Okay, great. Thank you, Franklin. Again, seeing no further questions in the queue, I'm going to thank everyone for joining us. Oh, again, just the last second question I just saw pop up, which we'll squeeze in here. This question is for Masi from Michael Del Castillo from the University of Saskatchewan. When you're looking at aerial images, did you encounter any problems regarding the calibration of the equipment used for the image acquisition? Did your data change due to light conditions?
Thank you for the question. For calibration, we use a tutorial existing on Micason's website. At some point, we had some problem with the images that capture the calibration panel. When it is not fully covered, the model actually failed to recognize it and failed to process the images. But eventually, we figured out that we need to change some metadata, and after that, the technique and the pre-processing tool was working pretty well. And after, we can find this problem and we solve the problem.
Okay, thank you very much for that answer. I will now try to close the session for a third time. Thank you everyone for joining us at today's session, and thank you especially to our speakers who have provided us with a very interesting panel. Our next live session will begin at 1 p.m. Mountain Time, but between now and then, you'll have a chance to interact virtually and ask questions with this year's poster presenters by dropping by their individual poster pages. To join a chat, look for the virtual table on the lower left-hand side of the page. This opportunity will run from 11 a.m. to 12 p.m. Mountain Time. Once you've had a moment to check out the posters, you can vote in our People's Choice competition via the link on the main poster page. So thank you again, everyone, and enjoy the rest of this year's symposium.